All right, genetics. So first, hearing. Okay, so think about it. We've got to the left, we have a Chihuahua. To the right, we have Golden Retrievers. Now, have you ever noticed we got a lot of different dog breeds? Okay, so how did they actually become to be? Now, we have actually created these breeds called through selective breeding. That means basically we get to choose the best trait. Okay, so this happens in most domesticated animals. Okay, dogs, horses. Now look at these 4 pictures. Okay, what do you think this top left picture? What do you think the best trait, or why we would want to choose that? So that's a thoroughbred. It has very long legs, so it's used for racing. What about the one on the right? So this is a Clydesdale. This is a working horse. Okay, so it's very strong muscles. Okay, it's used for pulling carts. And if you've ever gone to Busch Gardens, it actually is. You will see the Clydesdales there. They're also they will pull the carts. What about this bottom left? What do you notice about it? Has very long mane and tail. Well, we actually use that for cosmetics. Okay, for weaves. What about the picture to our bottom? Our last picture. It's polka dotted. Now, what would that benefit be? This is actually called an Appaloosa, and the reason why it's polka dotted is to help with camouflage. So the Native Americans would actually use these organisms to, yeah. All right, so even our cows and our sheep and our pig, okay, we actually have selected. They're a product of selective breeding. Okay, this is artificial selection. Now, our cows, our pigs, and our sheep are all used. The best trait is to make them bigger, grow faster, so we can actually consume them. All right, so hybridization. Okay, so you've probably seen hybrids throughout your life, okay, and you don't even realize it. So we already talked about dogs and cats, right? Okay, so we've talked about different breeds. Okay, so a hybrid is basically where we take 2 dissimilar individuals and we actually trade them together. Okay, so hybrid also can be hybrids can be different species crossed together. Okay, or it can be different types within the same. So we have talked about designers. Okay, so we have a golden doodle, which is this picture on the left. We have a puggle, which is this picture on the right. So is this considered a designer breed, or is this considered mutts? So we've got a pit bull. We have a pug. What do you think this new hybrid is? This baby is called a piddle. So, not to be confused with the hybrid cards, the eat. Okay, so these hybrid cards are a combination between using fossil fuels like gas and using electricity. So, inbreeding. Okay, so inbreeding is basically where you're going to breed with similar individuals. Okay, this is where we create purebred dogs. Okay, the problem with having purebred dogs is because you're not having the genetic diversity. Okay, because you're breeding with the same individuals, or the royals used to do this, right? They keep it in the family. This has a lot of risk to it, okay, because they're susceptible to diseases and deformities. So, for example, in golden retrievers, they have purebred golden retrievers has a higher chance of having epilepsy, which is caused by having seizures. Okay, so Dalmatians, purebred Dalmatians, they actually have deafness. That's why they're actually great firehouse dogs, because they don't need protection from the very high pitch fire alarms. So what's a mutation? Okay, so a mutation is any change in DNA. Okay, this actually causes a lot of increased variation or genetic diversity. Okay, so here's our Scottish fold cat. Okay, believe it or not, this was actually an accident. Okay, so this mutation happened randomly. Well, the cat breeders thought, oh, they're so precious. So these cats, okay, so the breed actually came from a single cat with a mutation for their folded ears. All right, so let's do a quick check. Okay, so dogs and other domesticated animals were produced through many years of selective breeding. Two dissimilar organisms crossed together is called a hybrid. Purebred dogs can sometimes have poor health due to inbreeding. Any change in DNA is called a mutations. Designer dogs, such as the Puggle, are also called mutts. All right, genetic engineering. Okay, so basically this is where we as scientists can actually change or modify DNA in any organism. So in our next unit, we're actually going to, when we talk about DNA, we're actually going to extract your own DNA, which is what this sample is. So if you look, this is actually DNA from a strawberry. So this top portion, okay, the clear portion where it kind of looks like strings or bubbles, that is actually the strawberry's DNA. All of this reddish orange stuff that is at the below, okay, that second layer, that's actually the strawberry's cells. Okay, so DNA extraction is where we're going to be able to do that later on in the year, later on in the term. We are going to remove the DNA from that organism's cell. So basically DNA sequencing. Okay, so we actually can use DNA sequencing, which is figuring out DNA's language. Okay, so basically there's 4 letters, okay, in DNA's alphabet. There's A's, T's, G's, and C's. Okay, so depending on their sequence or depending on their arrangement, okay, we can actually figure out and solve crimes, we can determine paternity, or we can even test for genetic disorders such as Down syndrome. So how do we actually do it? Okay, we use this device right here. This device is actually called gel electrophoresis, where basically we take electricity and we use an agar gelatin, which is basically made out of seaweed, and we create wells or holes. And we take the DNA separated by its size, okay, so the length of the sequence. And next slide, okay, here's an actual picture of it. We add electricity to it. And as you can tell from this illustration, okay, you can actually see the fragments moving or racing. Okay, so basically, the smaller the fragment you are, the faster you're able to win the race. The slower, okay, or the bigger fragment, the slower you are. So it creates these things called bands. Okay, these bands are actually called a DNA fingerprint, where basically we can take or we can see, okay, how these for this example, this is using bloodstain from a crime scene, and we're comparing to the actual potential suspects, okay, and their DNA. Now, what we do as forensic scientists is we're actually taking the bloodstain or the middle DNA fingerprint, and we're comparing it to suspect 1, 2, 3, 4, 5, 6, 7. Okay, and pretty much what happens is we have to see how many bands or how many segments of DNA actually align to our bloodstain. So the most matches is the winner. Okay, so which one of these suspects do you believe were this bloodstain belonged to? So the answer is number 3. So suspect 3 is the criminal. So why did we do this? Okay, so why do we go through gel electrophoresis, and how can we actually figure out, okay, using these bands and using all of our genetic information? Okay, we did this for a reason, okay? So it's called the Human Genome Project. Basically, it started in the 90s and it ended in the early 2000s. Well, the reason why we did it is because we actually were trying to figure out, okay? We were trying to figure out basically some clues, okay, on our basic properties of life. We were trying to figure out how genes can basically be used for commercial value, and also for development of drugs as well as treatment for diseases. Okay, so we took There were 13 different countries working on this for about 10 years. So now we know every single gene sequence in the entire human. Okay, we know everything. We know exactly the order, how the sequence, how the code is actually in every single cell of the human body. All right, so let's do a quick check again. So making changes to DNA is called genetic engineering. Gel blank is used to separate DNA fragments. Gel electrophoresis. Scientists can blank DNA from any cell or body tissue. Extract. A DNA blank can be read to determine paternity or solve crimes. DNA. All right, transgenic organisms. Basically, this is, we experience this every single day of life, okay? Transgenic, basically, trans means to change, okay? So it's where we take genes from other species and we insert it to another species. Okay, so we have species number 1, we take species number 1's DNA and we insert it into species number 2. Okay, so we do this in a couple different ways. So we use this with microorganisms such as bacteria, animals, mainly mice for medical uses, plants, okay, artificial uses. So you might have heard this term for transgenic organisms such as GMOs, or genetically modified organisms. Okay, so I want you to watch this film. And I want you to take a minute or two to go on YouTube and look for Jimmy Kimmel GMOs. Okay, this is where he walks the streets and he asks the citizens of California what a GMO stands for. It's pretty comical and a little scary that the public doesn't really know what a GMO actually is, or do they? So, GMOs are, this is an example, okay? So anything that is genetically modified, okay, that we have changed. So for example, we have these mice, okay? So what we did is we took the bioluminescent gene out of the jellyfish, and we actually inserted it into the DNA of a mouse, and it actually has the ability to glow in the dark. So these mice are real, and they are glowing currently right now. Okay, so let's move on to cloning, okay? You've probably thought about cloning at least once in your life, okay? Let's all be honest. It would be great to have somebody else go to school for you, or somebody else do your homework, or somebody else have to do chores, okay? So what exactly is cloning? So basically cloning is where you have organisms that have the same identical genetic code, okay? So twins are actually a natural clone, but clones can be made in the lab, okay? So we have actually done this. We have never cloned a human yet, okay? That is quite a controversial topic. I mean, it falls underneath the ethics category, okay? Is this ethically appropriate or not? Okay, so the very first clones were actually made through artificial twinning. Okay, so basically we take identical embryos, we separate them. Okay, they start to divide, and we implant them into 4 different surrogate mothers. Okay, so the identical are cloned that were once just in different mothers. Okay, so different holding areas, but they're the same. They're from the same cell. All right, so how did we clone a sheep? Okay, so the sheep were the first mammal that we actually successfully cloned. Anybody know what the sheep's name was? So her name was Dolly the lamb. Okay, so if we wanted to, and for Dolly, this was reversed. Okay, so we have a male. We take the body cell from sheep A. We extract the DNA from sheep A. We take the egg from sheep B, and we extract, or we remove the DNA. Then we insert the extracted DNA from sheep A, and we insert it into basically the shell, or the eggshell, really, of sheep B. Okay, it fuses together, and we're going to fuse it into until it basically starts developing into an embryo, and we're going to place it into a foster mother. Okay, so we actually the sheep or the lamb is an actual clone of sheep A, not sheep B. Okay, so sheep B was basically just a holding tank, really. So we've done this with sheep. Okay, so we have the technology to clone humans, but should we? And can you think of any reasons for cloning, any good reasons for cloning a human? All right, so we talked about it. Clones do exist naturally, okay? So these identical twins are an example of natural clones. So if you want to look more in depth about cloning, click on the link. So quick check. An animal that contains the gene of another species, like the glow-in-the-dark mice, are called a transgenic organism. Okay, or you can also refer to it as a GMO, or genetically modified organism. So number 2, a blank is a genetically identical organism. That is a clone. In humans, a naturally occurring clone is a twin. Okay, but what kind of twin? Identical or fraternal? It's got to be identical. The first mammal that was cloned from a skin cell was a sheep, and her name was Dolly. Dolly the. With regards to cloning, is it necessary to have which of the following? Check all that apply. Egg from the adult to be cloned. Egg from the nucleus. Egg with nucleus intact. Embryo, surrogate mother, sperm, stem cells, egg with removed. Okay, so we need cells from the adult to be cloned. We need a surrogate mother, and we need the egg with the nucleus removed. All right, so take a minute. Okay, we want you to kind of mull this over. All right, so we talked about, is it ethically appropriate for humans to actually clone themselves or others? Okay, so here's a couple examples. I want you to kind of mull it over and really think about, would you do this or would you not do this? Okay, and figure out some reasoning behind either to support yourself with your decision to say yes or your decision to say no. Okay, so what are some pros, what are some cons to each of these scenarios? Okay, all right, so I would use genetic engineering to remove a harmful gene from my unborn child, such as the gene that caused cystic fibrosis. Okay, if you don't know what cystic fibrosis is, basically what this disorder does is it makes your respiratory cells actually overproduce mucus, and basically essentially you pretty much drown from your own mucus. Okay, so would you do this or would you not do this? And think about some reasons. I would use genetic engineering to remove an abnormal but not necessarily harmful gene from my unborn child, such as the gene that causes dwarfism. So again, think about a pro, think about a con, and why would you do it or why would you not do it? I would use genetic engineering to remove a gene that is not desirable, such as the gene that causes baldness. Would you? Wouldn't you? And why? I would use genetic engineering to change a gene in my unborn child, such as their hair color or eye color. Would you? Would you not? So, I would use genetic engineering to add a gene to my child that is not human, such as the gene from another organism that could improve sight or running ability. So number 4 is actually possible. You do have the ability now, okay, for a hefty fee. You could actually choose your hair color, eye color, or even the sex of your child. So that does exist. Okay, I would use genetic engineering to add a gene, okay, that's not human. That's transgenic organism, or GMOs. Basically, we are making super soldiers like Captain America. So we're going to talk about Mendelian. Okay, so Mendelian genetics is very, very interesting. You probably have some prior knowledge on this. All right, so probability. Okay, so probability is basically if number of possibilities. Now Mendel, okay, Gregor Mendel, he actually is what, or we refer So he looked at pea plants. Now, he was a monk. Okay, so his job was to feed all of the other monks in the monastery. So he spent a lot of time in his garden. Okay, so he actually looked at pea plants because he could actually control their mating. Okay, so it was a controlled experiment that he conducted. Now, what he did is he would actually cut off the anther. Okay, so the anther and the stamens are the male natural. And so what he noticed is he took a purple plant and a generation. Okay, so what we did is we took a purple flower, a white flower, we mated them together because they're true bred. Flower color. Now, what he did is he took all the babies from F1, so all those purple babies, and he mated 2 of those purple babies together. And what he found out with their children, or the F2 generation, was that out of 705 the other. Now, let's do a quick check. Who is the father of genetics? Gregory Mendel. Genetics is the study of heredity, which is how traits pass from whom to whom? Parents to offspring. Mendel studied what? Organism. Pea plants. If one trait covers up the other, we say that it is dominant, and the one that is being covered up is the recessive. A true breeding plant is one that can only produce plants like itself. True. Okay, that's why it's called a true bred. If a tall and a short plant are crossed together, it creates a hybrid because it's two different traits. So let's alleles. Now, each box, okay, shows a different possibility, okay, or a genotype. So the phenotype is the physical So what he did, okay, is when a phenotype of a homozygous dominant and a heterozygous All right, so we've already talked about monohybrids. Now we're going to talk about dihybrids. So what Mendel really looked at, okay, so Mendel looked at what he found was this. Okay, so the F2 generation showed a 9 to 3 to 3 to 1 ratio, okay, for phenotypical combination. So let's do a little quick check. The passing of traits from parents to offspring is known as heredity. Who was the father of genetics? Gregor Mendel. Genes are located on chromosomes. Each gene is made of 2 alleles. You get one from mom, one from dad. The organism's outward appearance, such as wrinkled seeds, are referred to as pheno. Pheno is physical. Geno is the actual genetic makeup. So the letters capital R, that represents the traits are referred to as the genotype, because that is the physical makeup. An organism that has 2 different alleles or letters, such as capital R, lowercase r, are hetero, because they're different. An organism with 2 of the same alleles, like capital R, that's homo. Homozygous means same. Which of the following would represent Mendel's parent generation? Remember, they were true bred. Okay, so true bred, he bred one that was dominant, one that was recessive. Okay, so would it be homozygous or heterozygous for a true bred? So true bred, they're both homozygous. Okay, one is dominant, one is recessive, so that would be option C. When 2 different alleles occur together, such as capital R, lowercase r, what is actually the expressed? They're going to express dominancy. So what is this diagram called? This is a Punnett square. And Punnett squares show the outcome or the F1 generation from our 2 parent generations. So what does this letter actually represent? That actually represents 1 allele from 1 parent. All right, so we have a 1-eyed purple people eater crossed with a 2-eyed purple people eater. All of their offsprings have 2 eyes. Which trait is dominant? The 1-eyed or the 2-eyed? The 2-eyed. All right, so if you were using the letter capital E for this gene, what is the genotype of the offspring if the parents were both capital E, and lowercase? Okay, so if we took capital and lowercase, because we know which one is the capital. Is it the dominant or is it the recessive trait? So it's the dominant trait. So if any time there's at least one dominant trait, the overall expression is what? Dominancy. So all of these organisms or all of these offsprings would have the dominant trait, which according to our genotype, our phenotype, it would be 2 eyes. So if you cross, number 3, if you cross the offspring with each other, so if you could get capital E, lowercase e, and you times them together, how many of them would have 2 eyes? So if we times capital E and capital E and lowercase, 75% of them would have capital E, 75 of them would have 2 eyes, and 25% of them would have. So let's talk about pedigrees. Okay, pedigree, not the dog food. Okay, so pedigree is used to study sex-linked traits. Okay, so pretty much the males are indicated by boxes, females are circles. Now, if you are affected, okay, you are colored in. If you're not affected, then you're left blank. Heterozygous females are called carriers. So basically they might be showed half colored. Okay, so Alice is a carrier. Irene is a carrier. Fred has the disorder. She's a carrier. Now, let's talk about hemophilia. Okay, we call it the bleeder's disease. Now, if you are a male and you're normal, you're XY. Okay, for this case, it attaches the normal. Now, one of the most famous pedigree of all time is the English royal family. Okay, so starting with Queen Victoria. Now, Queen Victoria was a carrier. Okay, so if you look, Alice was a carrier, okay? Beatrice is a carrier, but her son, Leopold, he actually has the disorder. So you notice that each generation, each time, okay, at least one family member was affected by this disorder, and it was males that. Now, incomplete dominancy. Basically, what this means is that this is an exception to the rule. Okay, so that means that if you take a true bred, for example, Snapdragon, true mix colors. Now, Rowan cows. Okay, so what happened when you cross a true bred white with a true bred red? So we already talked about our blood type, right? Okay, so blood type A, you could be AA or AO. You can be, for B, you could be BB or BO. Okay, now, or for AB, you can only be AB. That's it. There's no other type of genotype. Now, O's, you have So for your blood type crossing, okay, so for this, you're going to have to be able to show. So for your blood type, you're going to have to show your crosses, okay? So if you're an AB, you're just AB. If you are O, you have to be OO. But for A and B, you don't know if they're homo or hetero. So you need to show the crosses. So what you're going to do is you are going to, so let's do this first one together. Okay, so you have A and you have B, and you're going to cross them. All right, so polygenic. Okay, so polygenic is basically poly means many. Lighter skin. Okay, so if you have all 6 dark alleles, in the middle of the spectrum. Okay, so now the position inheritance. Now, we talked about it when we talked about pea plants. Okay, so remember, seed color and texture on different chromosomes, but they have an it to the gametes. Now, some gametes, or excuse me, some genes, okay, that are located on the same chromosome tend to be inherited together. Okay, we call them linked. Us. All right, so let's talk about sex linked. Okay, so calico cats. Now, if you're a female and you're a black cat, you have both B's. Going to attach to it. So let's look at a cross between a black female All right, in females, you can be sex linked, okay? For female